Good afternoon, everyone. Thank you for joining us. My name is Marissa Waterman. I'm the Marketing Director here at the Academy. For today's webinar, we'll have our past president, Dr. David Vicari, serve as the moderator, and he will also introduce our speaker today. During the presentation, you'll be able to submit a question by clicking on the Q&A icon on the bottom of your screen. Before we get started, Dr. Vicari would like to say a few words. Dave, how are you today?
I'm good, Marissa. Thank you for the introduction and welcome everybody. We have a large group, shows the tremendous interest in this topic, as I am also very interested to hear from our presenter today. First of all, I want to thank the Academy's patrons, which you see listed on your screen right now. These are organizations which support the Academy. The organizations receive benefits in return for their employees and for the promotion of their businesses as well. If perhaps you belong to an organization that might want to consider becoming a patron, please reach out to Marissa at mwaterman@aaees.org. Let me now introduce our speakers, Dr. Daniel Yeh. Dr. Yeh is a professor in the Department of Civil and Environmental Engineering at the University of South Florida. He's also a visiting professor at the NASA Kennedy Space Center. Dr. Yeh's research and teaching interests are in water and wastewater engineering, global water and sanitation, the water energy food nexus, and life support systems for space travel. Dr. Yeh holds degrees from the University of Michigan, a Bachelor of Science in Natural Resources, a Bachelor of Engineering in Civil Engineering, and Master of Science and Engineering in Environmental Engineering, and from Georgia Institute of Technology, a PhD in Environmental Engineering. Dr. Yeh is a professional engineer and AAES Board Certified Environmental Engineer, or BCEE, and a lead accredited professional. As a BCEE and as a member of the Academy, I just have to mention, he's listed in the who's who in environmental science and engineering. Dr. Yeh served as the 2023 AAEES CAPI lecturer, traveling around the country and delivering lectures at various institutions. So let me, one quick reminder again, if you do have questions, please click on the button at the bottom of your screen that says Q&A, type it in there. I will collect the questions and put them to our speaker at the end. So with that, Dr. Yeh, please.
Okay, great. Thank you, Dave, and thank you, Marissa, for the introduction and for the opportunity for me to present today some of the work that our lab has done over the past few years. So I'm going to be talking about algae, and I'm going to be talking about membrane bioreactors today. This is the technology called ICARUS, and I'll explain what that means. It's a floating membrane photobioreactor that we can float directly on an impaired water, such as wastewater directly. The work I'm going to present today is largely the PhD work of Melanie Pickett, but also I'll be showing some of the work of her predecessor, Ivy Drexler, and also some of her successor, Gary Hsu, as well. Okay, so first, why the interest in algae, right? So from a planetary perspective, algae, different types of algae, microalgae, macroalgae, they're very critical to the balance of our ecosystem. They're primary producers, right? So they recycle carbon dioxide into biomass, which then could be incorporated into higher organisms such as grazers, other heterotrophs and so on, and ultimately into mammals and you and I. So they're very important in terms of the base of the trophic pyramid. The other thing is that they generate a ton of oxygen. Right? We would be in trouble without algae. About half the oxygen on our planet is generated by algae. So they play that very important role. But as you know, we often have harmful algal blooms as well, and ruining our water quality in coastal areas, and also drinking water reservoirs. And so, it's sort of there's the good, the bad, and the ugly related to algae. So it's a matter of they're critical. We need to manage them. But in recent years, or should I say recent decades, there's been tremendous interest in harvesting algae, right? Cultivating and harvesting algae to take advantage of the biomass that's created, which is usually comprised of different blends of carbohydrates, proteins, lipids, and other components that can be converted to a variety of products. So we can make biofuels from there. We can create diesel and it can be turned into jet fuel. We can make bioplastics directly from algae. They create precursors for plastic. So algae could be used as a way to recycle carbon, if you will, and we can make plastic instead of using petroleum, right? We can also take advantage of the proteins and other components of algae to make animal feeds. We can make fertilizer, but there's some really high-end products that we can make for nutraceuticals, dietary supplements, pharmaceuticals. One of the very expensive dietary supplements, astaxanthin. It's an antioxidant. It's quite pricey, and it's directly derived from the algae Haematococcus. So there's a number of benefits of growing algae, and one of those I want to highlight, because anybody who's traveled anywhere recently, you can't walk from from land side to from the air side to onto the plane, through the jetway without seeing advertising from the airlines about how they're embracing sustainability, how they're focusing on decarbonizing, and this term sustainable aviation fuel, right? So this is sort of like the path forward for airlines. They're going to switch from petroleum derived fuel and to sustainable aviation fuel that's derived from various biomass, including algal biomass. So there's definitely a tremendous interest, in algae from this regard. So how close are we to an algal-based economy? Well, there are some major barriers for sustainable algae production, and it has a lot to do with economics, right? Like everything else. So first, algae are, a lot of them are plants, right? There are exceptions, and they use water, nutrients, and CO2 to be converted into biomass. So very first, right off the bat, you're going to need water, and a ton of water to grow algal biomass. You're also going to need nutrients, right? Nitrogen, phosphorus, and other trace elements to grow biomass. So so we have to look at how available these are, and can these be provided sustainably, the water and the nutrients. The second is when you're growing different types of algal crops, right? There may be certain species that you're interested in growing. For example, some of them, have really high lipid content, or maybe you're growing chlorella and you don't really want anything else that's in there. So being able to maintain some type of purity, or monoculture or uniculture, is quite important. You don't want to end up with a mixed culture of just whatever, and that's one of the key problems right now, is that when you're growing them, say in outdoor ponds, which is the more economical way to grow them in some type of outdoor raceway, they're very much prone to contamination. And once they're contaminated, for example, with grazers that fall in there, you can decimate the crop. And the last thing is, how do you harvest and the algae, and how do you dewater the biomass? Because as dense as they get, it's still largely water, and there's water between cells, there's water within cells. So getting that water out is very important, right? To really end up with a thick paste and convert the products, and that's where a lot of the energy costs is. Right? There's a lot of different approaches and techniques related to that: belt press, centrifuge, conveyor dryer, so forth. But a lot of the energy goes into that. So we have to kind of overcome all of these things, right? So we need a sustainable source of water and nutrients. We need to be able to maintain their purity, and we need an economical way of separating the algal cells out from the water after we've grown them to a certain density. So here we go. Okay. In recent years, there's a lot of interest in some type of control algal cultivation and wastewater, right? And looking at the synergy between these two systems, and they're very complementary. For example, in wastewater treatment, if you have in some type of an aerobic process like activated sludge, you're going to need oxygen, right? And that's obviously a major cost factor on the side of wastewater treatment. Well, guess what? Algae as plants, they generate oxygen. So if we kind of combine the two, algae can create the oxygen needed for bacteria, right? So that's a nice synergy right there. The bacteria, upon converting, the BOD um, upon conversion of the BOD, generate a lot of CO2 emission. And this is one part of the carbon emission of wastewater treatment plants, right? The direct release of CO2. And in fact, the CO2 level, if you measure them in an activated sludge process or in a clarifier, it's quite high, it's super saturated. Right? So there's this source of CO2 that we can potentially harness and make available to algae, which requires CO2. Normally, we're going to have to think of where would we get a CO2 from, because we need more than just what's there in the atmosphere. So people are looking at, for example, power plants or cement kilns or other sources of CO2. But if we can couple this with wastewater, I mean, this is a sustainable, renewable source of CO2. So it can help, to work on our path to decarbonizing wastewater treatment plants. And then the last, obviously, is nutrients. There's a lot of nutrients that we spend a lot of time and money, effort, technologies on removing. And, those same nutrients are essential growth ingredients for algae, right? So, and of course, water. Wastewater has usually more water than we know what to deal with. We can recycle the water, but a lot of times that water is just being discharged. So it makes sense for this to be served as a renewable source of, feedstock essentially for growing algae, right? So here's the concept of doing that, but in actuality, it's more challenging than that because there are a lot of dirty things in wastewater, right? Namely, in wastewater, either raw sewage or the activated sludge process, there are a lot of organisms in there. And those are organisms that will either contaminate your algal crops or they will consume your algal crop. And, one of those is a group of organisms called rotifers. Right? These rotifers, actually, if you look at them in microscope, they're kind of cute, but they excel at just consuming, microalgae. They can eat thousands of them a day, and so if they get into your cultivation system, they'll decimate your crop. And imagine in the wastewater system, they're just natively present. So that's one of the challenges, in addition to all the other things in the activated sludge, fauna that's going to contaminate your crop. So in our lab, we work on membrane bioreactors of different types. We worked on, aerobic MBRs, anaerobic MBRs. And, when we start to think about this idea of how do we couple wastewater with algae, of course, our thoughts naturally gravitate towards membranes, right? Because membranes are these, incredible materials. Depending on the type of membrane you use, you can really selectively control what goes through by using membranes of different pore sizes or different materials with surface characteristics. And we can really control, what passes through. So, for example, if you use a looser membrane, right, like microfiltration or ultrafiltration, where water and ions are free to diffuse through, as well as gases, we can end up with this type of setup where, what if we grow an algal cell in some type of a sealed reactor of some sort? It could be a bag, it could be a compartment. We grow the algae in there, we float it on top of the wastewater, which has all the stuff that we want, but we put an in membrane in between as a mediator. So now water is free to go back and forth on an as needed basis. Nutrients are free to go through on an as-needed basis, right? And the other important thing is gases are allowed to exchange. So algae produce excess oxygen. They can now go into the wastewater and maybe provide benefit to the wastewater system. But more importantly, the feedstock for algae, CO2, can freely diffuse into this type of system. Any waste products could just be secreted through the membrane, and most importantly, anything that we don't want to be in there, competitors, grazers, anything that will contaminate or damage our crops can be kept out. All the other microorganisms, and this, we can put this on top of the water, right? So we get around these issues of self-shading that might happen when you have an outdoor growth system. Those cannot be very deep because you get through past a certain depth and the crop gets so thick, you start to have self-shading. So you kind of need to be at the very top to have access to the sunlight. So here's the idea behind the ICARUS concept. And ICARUS, you always need to have some acronym. So ICARUS is isolated cultivation of algal resources utilizing selectivity, right? And of course that that reference to the sun, from the mythology. Okay, so this is the idea, and then we think we can potentially put this on any type of dirty water environment, wastewater, or stormwater ponds, agricultural lagoons, and it's scalable, right? Potentially scalable. We can have different size reactors. We can combine them together, and then the important thing is we can generate high value biomass. And as I'll show, in the slides later on, we're able to really maintain crop purity in these things. So now this also opens up the idea that we can have different ICARUS pods floating on the same wastewater system, but in each pod, we can inoculate it with a different type of algae. So we can simultaneously grow different crops in the same system without cross-contamination. Right, so it's basically growing algae on a by order, on an as-needed basis. Right, so these are all the ideas, but like, how do we make this actually happen? So we draw inspiration from NASA's technology readiness level. So this is basically a way to benchmark technology development, starting with TRL 1 all the way to TRL 9. Right, so TRL 1 is the most basic level. You have an idea, you want to test it, with some type of just really quick proof of concept, lab on the lab bench scale, simple component, not even on a system level, and then you start to kind of get more information and move up the TRL levels, and by the time you get to 4, components are coming together as systems, and when you get to 5, you're testing your system in a simulated environment. It can still be in the laboratory, but at 6, it should be in some type of operational or relevant environment. So, and then 7 is when you start to scale, you're going into a real environment, right? So in our case, this would be actually, say, deploying this at a wastewater treatment plant, being exposed to actual wastewater, rather than doing this in the laboratory and using either synthetic wastewater or bringing wastewater back to the lab. This would be actually in the lab. And then you start to go through further maturation until when you get to 9, you're ready for this to be deployed and possibly commercialized. So we started with the very basic TRL 1, right? And this was the work of Ivy Drexler, where she kind of tested out some ideas. And we just started with dialysis bags, right? Which we know that ions can diffuse through dialysis bags, but the pores are small enough that algal cells could be retained. If you look at the picture above, we tested chlorella species, Sorokiniana, and right here, this is just a dialysis bag, and more like a dialysis tube that we clamped, and you can see we can grow algae in here being submerged in a nutrient solution, and it's green inside. We have cell growth, but there's no leakage. So kind of like we can start to kind of get on this journey of creating an isolated system of growing algae within a bag, right? So some of the bags, because the bags are very flimsy, they're ripped. So we knew that as we go forward, we need to have something a lot more durable than dialysis bags, because they really don't have a lot of mechanical strength. And so in the second, moving on to TRL2, we started to think about how do we create a component of some sort, or a mini reactor, right, beyond a bag. So we also need something that's reproducible, so we can have a lot of a series of a lot of these pods that are all the same, but maybe the only thing that we're changing are the different types of membranes on the bottom. So we use these kind of PVC fittings that we modified and put a membrane on the bottom, and then essentially we can then put this on jars, or have these open top systems, and we created an indoor raceway, with lighting. So now we can create we can fill this in sort of a nutrient system, but simultaneously test different types of membranes. That's the first thing where we need to find out what membranes work well, so that we can down select. So by doing this, we're able to screen through a number of membranes, and we tested a variety, regenerated cellulose, cellulose acetate, nylon, PVDF, and number of sizes. And we found out that somewhere between 50 and 1,000 kilodalton was the best, right? So it allows us to kind of down select a little bit. And then we said, well, why don't we kind of take these pods now to actually the wastewater treatment plant, right? These are obviously not at the level of the reactor that we want, but we have these reproducible cells. Let's just put them in the wastewater. So we went to Tampa's wastewater treatment plant, suspended baskets of these pods in the clarifier, and let them grow over about a 20-day trial, and with these different sizes. And we had some kind of pleasant surprises and some not so pleasant surprises. We found out that we're able to actually grow much higher density in the field than what you observe in the lab. And the reason for that is simple, more intense sunlight than artificial lighting in the lab. And also in the lab, you have a limited feedstock in the control system, whereas in the field, it's an endless supply of nutrients, but more importantly, carbon dioxide, right? There's tons of carbon dioxide in the clarifier, constantly fueling the algal growth. And one of the problems we ran into is that there's so much bioactivity that in some of our pods, they dried out. I mean, we had trouble sampling because, when we went back to sample, water, basically we end up with like a paste in there rather than, suspended culture as we wanted. So that made sampling difficult. And then, so here's some data from that part of the trial. The 4 grams per liter is something that we were benchmarking, comparing against, because that's a typical raceway concentration for outdoor raceway systems. And here's basically a series of either blanks using the small, medium, or large size membranes. And we found out that ultimately the one that grew the best, able to achieve more than 10 times on a grams per liter basis, was the medium-sized membranes with the PVDF membranes. So that did the best. Nylon is also interesting because it's very cheap, 7 microns. But what we found out ultimately with nylon was that it was just too leaky, and also it allowed other things to get through that we didn't want, which I'll talk about later. Okay, so this was sort of really a proof of concept that shows that this idea could work. But obviously, there's a lot more technology development and engineering needed to get this into some type of a reactor that actually would work, and how do you actually scale this up. Okay, so future research coming off of Ivy's PhD work was that we need to really think about how nutrients and gases exchange in these systems, demonstrate crop protection, right? Because here we're just looking at density of algal crops, but we're not really, really characterizing how the purity of the culture. So we need to demonstrate that, and we need to understand a lot more about membranes foul in these systems, so that transitions now to Melanie's work. And here we envision TRLs 4 to 5. Under TRL 4, we're going to start to test multiple components together. We need to understand better membrane diffusion, crop protection, membrane fouling. And then we're going to move on to 5 and 6, simulated to operational environments. started to actually develop prototypes, beyond these like, baby jars with a membrane on it, and start to move, using these prototypes to test them in these actual environments. So that was the scope of Melanie's PhD work, and these are all the different things that we want to figure out, that I just mentioned through the research coming up. So the first thing was need a better understanding of mixing. For example, in these baby jar systems, there isn't a whole lot of mixing, right? But as you kind of scale up, we can imagine that mixing is going to be important in these systems. So we created now bigger cells. These are actually like plastic bins you can buy from places like Walmart, but they allow you, but they're sealed reactors, and you can easily manipulate them. And we were basically testing the effects of different types of mixing on growth, and how that improves growth. And the short of it is that mixing, of course, as you would anticipate, led to increased growth. And so we got information from that, and of course, if you have a membrane, it starts to foul, and backwashing that membrane might be important. But then, the challenge is going to be like, okay, we have these cells in there. How do we actually induce mixing? And more difficultly, how do we actually generate backwash in these cells without making the system overly complicated now with, pumps and other components, in there, right? But, so we take that idea moving forward. And then, recall that in TRL3 trial, we had these jars that really ballooned out, and it caused us to lose the sample. It was very difficult to characterize really how much algal growth we had. My father always told me that, just when you have something fail or not perform the way you want it, it's not necessarily a failure per se, right? It could just be that you're going about it the wrong way, or maybe you need to reset your expectation, or maybe what you're working with can really be better applied to something else. So, we stare at the system and try to, think about what caused the loss of fluid in these systems. And it dawned on us that, algae do photosynthesis, right? I mean, the equation is, oxygen, or I'm sorry, CO2 plus water leads to biomass and oxygen. So oxygen is the product of photosynthesis. And if you think about, yes, it also consumes CO2, but it generates oxygen, but CO2 is so much more soluble in water than oxygen. So basically, there's a net production of gases in the gas phase in the system. And that's why it dewater. As the algae was doing photosynthesis, headspace pressure built up to the point that it pushed water out of the membrane, right? So this was a failure from a perspective of doing batch experiment, but it's a success if you think about what's going on here. It's a self-dewatering system. Remember what I said earlier about the three major costs with algal systems, and one of them is dewatering? Maybe we're onto something here. Maybe we have a self-dewatering system that could happen, right? So, you're growing the algae, right, in the system, and you get into this phase, where oxygen starts to accumulate, right? So what if we can regulate that pressure? So basically, while the algae is growing, we allow the excess pressure to leave the system so that we don't lose the fluid, right? Water can still come in. And then, when we're ready to harvest, we close the valve, right? So basically, you can have a very simple valve on the system, like some type of solenoid valve or something that could be very easily controlled, either by time or by pressure. So during the cultivation stage, we let it grow, we let gases out. But then when we're ready to harvest, we close that valve, allow pressure to build up, and we push all the water out on its own without expending any energy. The process of photosynthesis will do it. Right. So this is the concept, and we went up to draft up some of these ideas. We have a couple of U.S. patents on this, but it's I think this is just matter to me. It's an example of look at failure, in a different way. It's maybe failure can actually lead to a different outcome if you just redefine what your objective. Okay, so we're gonna move on to crop protection now, the other objective. And oh boy, there are all these things in wastewater, right? It's really not just a system of bacteria. And those guys, rotifers, are certainly present. So we want to demonstrate that we can protect our ICARUS system from rotifers. So in our laboratory raceway system with the pods, same pods that we used earlier in TRL2, now we intentionally spike the solution outside the pods with rotifers. Right, so we went and bought a bunch of rotifers, and usually people buy them to like feed shrimp, right? So we bought them to basically challenge our algae system. So think of like these systems are like castles. You have algae in there that you're trying to protect against the rotifers who are trying to storm the castle. They're like barbarians trying to storm the castle. And the question is, can the castle gate, the membranes, hold up against these, rotifer barbarians trying to get in there? Right? And then we tested with an 800 kilodalton membrane. We did test it with the nylon, and nylon didn't work. Even though the rotifers are technically larger than a 7 micron, they snuck in somehow. We think it probably with their juveniles, they just got in, while they're small. So the point is that outside the ICARUS pod, right, you have rotifers in the order of 10 to the 6 number. And inside the ICARUS pods, after we did grow outs, we did not observe any rotifers inside. As you might expect, but we didn't demonstrate that. And of course, if you have a rip of the membrane, then you have a chance for contamination. But if the membranes were kept intact, they were fine. And over and over, we observed that not only do you not have contamination from outside, if you start with a monoculture and you keep the system sealed, you will end up with the same monoculture at the end of the grow out, and directly growing in wastewater. Again, very dirty environment. We did some investigation on membrane fouling using different tools. One of the tools you use to look at biofilms is the confocal laser scanning microscope, where you can use different types of stains and be able to look at what's growing on it. Are they live or dead, and what organisms do you have on there? This membrane system with the algae, with the ICARUS, is very interesting, right? And very, very uncommon in that normally when people look at biofilms on membranes, they're only looking on one side. Like for example, if you look at membrane fouling in a membrane bioreactor, you're looking at the side of the membrane that's facing the bioreactor, because that's where all the action is happening. People don't usually look at the other side, the permeate side. In this type of system, we have biology on both sides. We have the top side, which faces the algae, and we have the bottom side that faces the wastewater, all the bacteria and everything else that's in there. So basically, it's a duplex biofilm system. It was very challenging to actually preserve the membrane and get them under the confocal without, because without damaging one side or the other, But anyway, so this is the type of stuff that I am, keen to work with people who have a lot more experience than we do, and see how we can move this forward. Not only further characterizing this duplex membrane, but actually develop rigorous mathematical models on what exactly is going on in this type of duplex biofilm system. Okay, so it's summarizing sort of the lesson so far. In TRL1, we were able to show that wastewater could be used as a growth medium, and we down selected it with TRL2. In 3, we demonstrated that in the field we can get additional biomass yield, and then something's going on with the gas regulation that can potentially lead to basically dewatering, self-dewatering. And in 4, we did some further investigation of mixing, the requirements for backwash, and demonstrated crop protection. So now we're kind of need to move on, beyond these kind of simple pods or bins to something that might represent how we're going to grow them, because these systems that you see here are fine for batch studies. but there's no way you can use these ultimately for large scale cultivation, right? Because it require opening up the jars or opening up the bins and recovering the algae. And here you see the students are manually harvesting these, for a science phase. But we're going to actually, scale this up. We need this harvesting to be automated somehow. And so we're thinking like, maybe we need to modify the geometry of the system so that harvesting could be more automated. And what we really don't want to do is have to take these pods out every time we want to do harvesting. Obviously there's weight. You have to handle this. It just increases the cost of everything. The complexity. We want to be able to leave these pods in the system, and just be able to grow them in place, right? So some of our requirements that I think about is so what do we have? We're able to flow this in impaired water, get nutrients from impaired water, get high density growth. We're crop protection. Maybe we should start to make the bottom tapered so that as we're growing, the algae can just kind of fall down the membrane towards maybe like a trough, right? And we have to create some type of a shear, something to encourage the excess algae to kind of go down. So kind of came up with this idea. Basically, have membranes on the bottom and sort of a slant, and so therefore you can maybe utilize gravity and encourage the algae to go towards the middle trough. And so this would be one cell, but you can potentially have a series of these cells that can tie together, right? So you can couple them together, and then the algae goes into this trough, higher into a slurry, and we can kind of pump the slurry out from a series of pods, forming arrays in a water system, something like this, right? So this is the clarifier, and it's a rectangular clarifier used in the city of Tampa, and we can put our pods in here, and if you want to think about a wastewater treatment plant, the first thing is you want to introduce the algae in a way that does not impede the function of the wastewater treatment plant, right? Because the main purpose of the wastewater treatment plant is to treat wastewater. Growing algae is a secondary thing, and some plants we talk to, well, they really don't want to hear about it because they don't want that distraction. And some, we're potentially interested in growing them. So basically, we did a National Science Foundation ICARUS program where we talked to a number of wastewater plants to find out what their needs and desires are and pain points. And so you have to have a technology that's in there, not very invasive, right? So we think about, also where do you have the largest surface area in the plant? Usually, is the clarifiers, and the clarifiers serve really one function, is to clarify the wastewater, separate the sludge from the water. What if we can just kind of park these pods on there without impeding the function, the clarifier to have overflows, right? And now we can basically tap into an underutilized real estate at the wastewater plant. So that's kind of like the idea. And so we started to make these prototypes, and we're like, well, how do we kind of create these different shapes, because we need to go through a lot of iterations, rapidly go through iterations of prototypes and see what works, what doesn't. So we decided to use this technique called vacuum forming, where you can build different size molds, right? And because we are testing different geometries and so forth. And in the vacuum forming process, you essentially have sort of like an air hockey table that you can make out of a pegboard. But on the bottom of that table, it's connected to a vacuum pump, like a shop vac, right? So you're drawing pressure down, and then on the top, you can have a sheet of plastic that now you need to heat up, right? So it's like, really, really soft and pliable. And then you just kind of pop it over your mold, and instantaneously it will conform to the shape of the mold because it's being pulled down by gravity. I mean, by vacuum, and you basically vacuum form and it cools, and now you have a sheet of plastic in a mold shape. And on top, basically, we just use space heaters, that you use, when it's cold. But we defeated the safety cutoff on the space heaters. Otherwise, it wouldn't get hot enough. So obviously, you have to be very careful when you're working with this. But yeah, and it took a lot of trials because you had to get your technique just right. And we had like haul of failed prototypes because we couldn't get the timing right, the temperature right, and the prototype just formed in a very, very ugly. But finally, we got the technique down. We're able to kind of make these prototypes repeatedly and generate a whole bunch of, top sides, bottom sides. So, the top side is really just, it was a mold, basically like a glass, yeah, mold, for use in the kitchen. So, all right, so we made these prototypes and we went through phases of testing them in a lab, in an outdoor environment, and in the field. So I'll quickly go over that, where we did there. So in the lab, it's still relatively simple. There we put them inside bins, but here we needed to more carefully characterize the performance of the prototypes. What's going on with the DO level, pressure buildup, number of things, and we had jars also floating in there as controls. And so here's how we did the setup. I'm gonna summarize the results from the lab, outdoor, and field, in one slide later on. And in the lab and the outdoor environment, okay, so now we went out into, basically in a large outdoor tank. We're able to have this actually be exposed to the sun and weather and all the good stuff. And we float a whole bunch of prototypes at the same time. And on the outside, we use a control simulated wastewater, which is just really miracle grow. We're just interested in can the nutrients get into the pods and so forth. So create a whole number of series, floating these pods outside, outdoor for the first time. And then for the field testing, we went back to the wastewater treatment plant. Sorry, there's a little bit of a delay here. But one thing we didn't have in that outdoor system is there was no flow, right? I mean, they were just floating, quietly in this tank. So we don't have the means of, tangential flow, replenishing the feedstock, all the stuff you would have at the wastewater plant. So, that's why you need to do the testing there. And at the wastewater plant, with the upwelling of the clarifier, you're going to have these constant tangential flow across the membrane. Right. So so this is what we set up. We set up, kind of like a cage, like a crab cage that we built. We put the pods in there because the number one thing is we cannot lose our samples into the clarifier, both from the perspective of our experiment, and we don't want to drop anything in the clarifier that's going to get the treatment plant angry at us. Right. So we had these cages where that safeguarded the pods or the prototypes in there, and we set them up at the wastewater plant in this actual environment and did a 1 month grow out. And again, in these clarifiers, there's constant upwelling of the water, providing an endless supply of nutrients and CO2 and all the good stuff. And the cells went in there like this. And here's another good shot of the clarifier. You can see, this basically, the water here just constantly upwelling, and spilling over. Okay, sorry, let me get through the slide. Here we go. Okay, all right. So here we, summarize the, from lab to outdoor to the actual wastewater treatment plant. When we have prototypes, jars, and controls. Right. And we look at dry weight in the reactor, but the other parameter is areal density. So basically, we normalize our production to the footprint of the system of the reactor, right? Because we want to be able to have even greater areal density than what's conventionally out there. So that for the same amount of space, you can grow more algae. And that's what we ultimately need to do because space, land area is always going to be limiting, right? So you want to grow as much as you can using a certain land area. You want to intensify that process. And then so density is productivity, and we're also looking at kinetics. And so in this column, but let me draw your attention to here though. This is what the summary is that for the prototype outdoors, you can compare it to the jars, to the controls. It was obviously better in every count. Our aerial productivity was 10.9 grams per square meter per day compared to the jar system, which has the same membrane on the bottom, but just in the jar. And we have higher kinetics and a number of things. So it does work, but the next issue is we still need to overcome a number of things. So in summary here, we achieve our highest growth ever in our trials at this time with using the clarifier at the wastewater treatment plant. And some more comment on membrane fouling. We kind of went back to look at the membranes and try to characterize that a little more. Using FTIR, we had Ken Ishida from Orange County Water District help us out in this regard. He's one of the world's top experts in this. We're trying to identify what in the world is growing on here in terms of the proteins, the carbs, the polysaccharides, and so forth. And what was growing on the different sizes of membranes. And here's some SCN images we also looked at. Actually, there's not gonna see a lot, but you can just essentially you're seeing the different caking form on the two sides of the membranes. And this is what the prototype looks. After spending a month in the field, you can see it's quite weathered, right? So if you scale this up, you're going to have to overcome a number of challenges, including durability of the membrane. And I think there's obviously economics. We have to get the cost of these pods down as low as possible, but make them as durable as possible, the membrane as durable as possible, and be able to basically do this backwashing. You're going to have growth on the membrane, and that's not necessarily a bad thing, especially algal biofouling on the membrane. You just need to be able to backwash that and get that into the trough, right? So that's where I think the future development is going to be. That automatic valve or that regulated valve is using that pressure difference to generate backwash, right, on a time basis, so that we can constantly slough off the biofilm that grows on there and collect all that biofilm as basically algal product. Okay, so then so basically some additional characterization of what is growing on the membrane. Okay, so the overall conclusion of this is ICARUS does work, but further development obviously is needed. We need to move beyond individual cells. We need to now go into an array of cells. This upwell of water in the clarifiers works really well. Here we tested in a rectangular clarifier. We have yet to test it in a more conventional circular clarifier, but it might work something similar. But there's this supersaturation of CO2 that I think people often underlook. We just focus on the aeration requirements for activated sludge. We focus on biomass and of course BOD removal and the CO2 just emitted. And it's a source of CO2 and it's a source of carbon that we can capture. So again, like I mentioned before, if we capture this free source of CO2, we can reduce the carbon footprint of the plant, and all the CO2 is free, right? So in terms of kind of future work, moving past Melanie's work, is we need to basically improve the process in a number of areas, increase the diffusion, backwash, manage the fouling better, durability improvement, move on from cultivation to harvesting. Now it'll be interesting to see how the cultivation keeps up with continuous harvesting, removing from the pods. And the idea is that we'd like to be able to leave the pod in there as long as possible. I mean, for weeks, months, even years if possible, so you don't have to remove them, and just really grow in situ. And maybe moving beyond wastewater treatment plants. This technically can work in different types of systems with nutrients. So agricultural ponds, stormwater ponds. There's areas like in Florida where we have a lot of ditches, canals that have just harmful algal growth. So instead of maybe growing in there, having some species that you don't want growing uncontrollably, could we line these canals with ICARUS pods and grow that type of algae that we actually want using the nutrients that are in there already, right? And potential application for space. Our lab is working with NASA, and there's also these opportunities to grow algae that you might need for space. The reason you want to grow algae for space is that obvious synergy that they generate oxygen that the crew can use, but they consume CO2, which is a waste product in space. But you can also grow algae for different products. I don't think we're going to necessarily make like rocket fuel per se, but and I don't think there'll be enough biomass for that. But grow beneficial products like nutraceuticals, specifically antioxidants, that the crew is going to need to fight radiation sickness. And this is something that you can't really stockpile very well. But if you can grow algal species to generate antioxidants on a recurring basis, now we start to pave the path towards long-term human habitation of outer space. So we did look into the idea of using ICARUS maybe in an aquaculture application, right? So can we, you think about aquaculture, let's say tilapia, right? You grow a lot of fish in a very small area, and the fish generate a lot of ammonia. So you need to remove the ammonia. And of course, the other thing a fish needs is oxygen, right? So can we provide the same type of approach, float ICARUS pods on top of an aquaculture tank, if you will, right? And actually be able to grow algae, which would naturally grow in an aquaculture system. And you were trying to manage that, and then they started to harm the fish. But can we selectively grow the algae, a certain type that we want, and be able to just harvest that? So Gary Hsu looked into this for his master's thesis. Interest of time, I'm not going to be able to get through all the work, but I want to show you some slides of the systems he built. So in the control system is basically tilapia growing in just in a fish tank, right? And this one has a conventional external nutrient management that you have. Like in the typical aquaculture application. So the fish was very happy, water quality was very good, ammonia was kept low. Compare this to maybe a system that you spike it with algae, right? Without some means to remove, they will look like this. But the same amount of algae that we spike in the system, if we put them in the ICARUS pods, will look like this. So now that same massive algae is growing in these floating ICARUS pods directly in an environment that we're growing tilapia, using the nutrients released by the fish to grow algae, but exactly the type of species that we want, and generate biomass that we can harvest on an ongoing basis to help out the fish, but derive a secondary product for aquaculture. And you can, generate different types of algae, but perhaps even use it as fish feed later on in some other system. But the thing is, if you try to, grow algae for fish feed, well, you're starting to get into some problems for the fish with, clogging of the gills and so forth. So yeah, so this is work for another presentation based on Gary's work. But to wrap up, the work of Melanie Pickett's, which is the bulk of this presentation. So here are all the people who helped out. We basically had generations of students working through 3 graduate, 2 PhDs and 1 master's thesis, all related to ICARUS, and all these students worked on it. Tampa's Howard Current wastewater treatment plant was tremendously supportive and helpful, providing us the location to actually test the ICARUS in the actual wastewater system. And then, of course, Ken Nishida from Orange County Water District. Melanie had a chance to go to University of Exeter, collaborate with some collaborators in the UK working on algae through an NSF opportunity. But most of the work here that I just talked about was funded through NSF. But starting with just a unsolicited seabed award, and later on moving to ICOR, and also this thing called AIR-TT, which is Accelerated Innovation Research Technology Transfer. So this was all built on NSF support. So with that, thank you. And I guess I'll take questions now. Back to you, Dave and Marissa.
Daniel, that was very interesting. I was very fascinated. Partly because of my own history, I also worked with NASA several decades ago on bioregenerative life support systems. And boy, if you need a circular economy somewhere, it's in a spaceship where you've got a crew trapped for about 3 years on a Mars mission.
Yeah.
And that it's great how you are able to bring those lessons down to earth in a literal sense. This is really terrific. Thank you. So let me try to summarize a few of these questions. They all kind of came in very quickly at the end, and there are a number. Um, one question has to do with gas exchange with the water. Does it only affect the top of the water? Also, is there much exchange with the atmosphere? How much goes into of the oxygen goes into the water versus how much into the atmosphere?
Well, there's no direct exchange with the atmosphere, right? Because it's a sealed reactor. The only opening is the membrane, semi-porous, which is in contact with the water. So the first exchange is into the water, and the second would be, I guess, from the water back out to the atmosphere.
Okay.
We didn't characterize that part. I have no idea what's going on between the liquid and the atmosphere.
Okay. There was a question about biofilm growth on the permeate side. Now, you did address scaling or just fouling on the, I guess, the permeate side. They mean the outside. What would that imply in this case? Yeah, that would be the outside, the clarifier side rather than the algae side. But you did look at that, right? You looked at the fouling on both sides of the membrane, didn't you?
Yeah, we did, and I think we're just really scratching the surface. I think there's a lot more. I think both, I think a lot more science, basic science that's going on in the system, in sort of this engineering system, and yeah, I'll be really keen if anybody in the audience is interested, and then they work explicitly on biofilms, either characterization or modeling, to
Yeah.
maybe collaborate on this. Yeah.
So preliminarily, did you find that it was very adherent to scale? If you hosed it, could you remove a lot of the scale on the outside, or is it something more adhering than that?
It wasn't very tough, I mean, but obviously our experiments were less than a month. I mean, you can remove that pretty readily, and it was just all sorts of gunk, like all sorts of biomass and bacteria and whatnot. But it would be interesting though if this is left on for weeks and months. Yeah, how tough that becomes, and actually getting inorganic scaling potentially, in this system.
Mhm. A couple of questions related to some practical details about using the wastewater treatment. One question mentioned about floatables. Will that affect your system and clarifiers? And then the second related question is whether this affects hydraulic loading rate on the clarifier, the overflow rate, because you're covering the surface partially. What do you think what effect that will have on performance of the clarifier?
Yeah, and that's a good point. We did think about that. I think the amount of flow that's coming up is so massive that it would seem to me that even if we cover it, the force upwelling force is so strong, it's not like we're not going to block the clarifier, if you will. But I think that it requires further investigation, and obviously you want to make sure the size that you have is not going to come anywhere near that. But from what we've seen, just the upwelling force is so strong that I think you can fulfill both functions.
If you don't mind me addressing that too, because I'm kind of a clarifier specialist.
Okay.
And
Yeah,
so I would say that most of the particle separation occurs in the water column and not at the surface. So you still have that surface loading, that upflow velocity throughout the water column. The particles will settle through that water column into the sludge blanket. I would not expect a severe issue there.
and that floatables is interesting. Yeah, that would be interesting. Like this, the various debris hitting the membrane over time, possibly damaging it.
Besides looking at biomass growth, did you look at nitrogen and phosphorus removal rates?
We did, yeah. And I will say right now, the biomass production is ahead of the nutrient removal. We are removing nutrients, but not as much as we would like. but definitely generating a ton of biomass. So you always want to think about, what is this system for? Is it for, I don't know, taking advantage of CO2? Is it generating biomass, or is it removing nutrients? Or is it all of the above? And I think right now it's not optimized for nutrient removal yet, or to be significant, but we could make it like that. I
Yeah.
don't think this is optimized yet for that. And I
Any,
think obviously, if we can harvest on a continuous basis, that will provide more sink for nutrients to flux across the membrane.
yeah. Any other water quality effects that you might anticipate? Even CO2 removal affects pH. It would raise the pH, but of course that might be supplemental CO2 to begin with.
Yeah, let me say something about that. I forgot. Normally when you grow algae, right, in any type of bioreactor, pH will go up because the algae is depleting the CO2 in the system, and pH starts to go up to a very uncomfortable level and can get up to like 9 or 10. In our system, because of that constant supply of CO2, the pH in the pod was kept very near neutral because of that constant availability of the CO2.
Any risk of blue-green algae growth? Is blue-green, and in fact, might that not even be a benefit? Could you run this? What type of algae did you use? And of course, blue-green algae are not algae, they're bacteria. So cyanobacteria also could, I suppose, be used, or what are the merits of some of the different kinds of cultures?
Yeah. So obviously algae grows where it wants, right? If you go to the way plant, algae is growing everywhere, and so including blue-green algae and other species. So a lot of people are interested in taking advantage of that. If you're trying to use some type of bacteria and algae combined process to remove nutrients from the wastewater, you would start with the algae that's already growing there, whatever it is. Obviously, it thrives in that environment. So our application is more about being able to grow selective strains that might be higher value that you want. For example, we grew chlorella. It's commonly grown for anything from fuel to bioproducts. They're very versatile and they grow very fast. So we did that. We also looked at Botryococcus brownie, which is relatively slow growing organism, but it has a very high lipid content, and people are interested in using that for biofuel. But of course, because they grow slow, you can contaminate them easily. So yeah, so we're able to grow different strains, and the possibility is obviously endless. But the idea that we can have like a grid of just different types growing all in the same system. I mean, I find that very interesting.
You haven't tried cyanobacteria yet?
No.
Okay. Another thing that I learned when I was working with NASA, I went to visit Disney World's wastewater treatment plant. Have you ever been there? It's right near you. And at the time, I don't know if they still have this, they had ponds after the final clarifiers where they grew floating water hyacinths, which they then mulched and put into the digester. So then you got energy capture from the sunlight and the carbon capture, and it was turned into methane, which would then be a captured energy. Are you familiar with that? And have you thought about using, besides have you thought about that benefit, ultimate use for your algae, and what other ultimate uses for the algae more directly have you considered?
Yeah, we so in addition to this type of floating algal membrane bioreactor, we've also done more of membrane bioreactor with algae in it, a phototrophic membrane bioreactor. That's more like a conventional MBR type. So basically, instead of activated sludge, it's just an algal culture growing in there. In that one you're using a pressure-driven membrane for that. Yeah, we've looked at different combinations, of especially anaerobic systems and algae, and plants obviously can play a big role in that. A lot of people are working on this. Nature-based solutions are very, very hot right now. Yeah, and I think all of those are great ideas, including there's several companies basically having using algae as a final step to after just some conventional wastewater treatment. for either deriving algal products or sending it back to the digester for more energy. So I think what we're doing here is more of a special application that, to basically crop purity is what we're after, that's these other open systems won't be able to have.
Yeah, speaking of the water hyacinths, one of the limitations of that was that they're not hardy. So I'm sitting up
I
here
have
in
not
New
worked with
Jersey.
water hyacinth, but I'm very intrigued with duckweed.
Okay, but so how about your system? It's covered after all. How far north do you think you might be able to apply your system? As I said, I'm here in New Jersey. We have nice spring weather today, but certainly January and February would be tougher.
Yeah, I think as long as the water doesn't freeze, there's a potential application. Obviously, it will slow down quite a bit. Well, you might be surprised when we did that outdoor testing. We actually did it during winter in Florida, and it actually got so cold that in that tank you saw, we had some ice forming on there. And they're like, "This is Florida. It's not supposed to happen." But
Might have, must have been welcome a cooling for you guys down there at that time.
yeah, well, it
Let me
plants
see.
when that happens. Like your landscaping plants.
Any preliminary consideration of cost benefit or economics for this? I'm guessing not at the TRL level you're at, but what would you say to that?
Yeah, for Gary's aquaculture work, we did some very preliminary TEA, but it is not at the level that we need to, both in terms of the LCA and also TEA. I think the main costs are going to be the fact that, if you have a raceway, it's very little moving parts. It all is all open, and now each of these pods are going to be costs. And then while it's growing, it's relatively free of energy, but we do need to kind of pump out the slurry, so there is that cost for conveyance. So I think it's going to be sort of a trade-off between having pods that are very durable and that might have higher costs to having pods that are cheaper but not as durable.
Mhm. And you probably know there's a lot of interest in the problem of nitrogen oxide emissions from activated sludge plants, especially nitrifying or denitrifying plants. Is there any potential for that, for such emissions from this process? N2O, which is a greenhouse gas.
Yeah, not sure. Because we're not, actively nitrifying these systems. That's not to say that the algae won't attempt it, but but whatever might be already happening with nitrous oxide emission is going to be outside the pod. I think most of what's going on inside the pod is just the algae is taking up CO2 for converting to biomass.
Mhm. I think you did mention that someone asked if this could be used for nutrient removal in lagoons.
Right.
That's a yes, right?
Yeah. Yeah. I haven't done that yet, but well, it's funding and time. I think so. Anybody who's listening is interested, happy to identify collaborators and just scale this up, find more applications.
Mhm. One attendee asked you to repeat the meaning of the acronym ICARUS for you.
Okay. It's isolated cultivation of algal resource utilizing selectivity. At first, we had the term sewage in there, utilizing sewage, but the industry was sort of like trying to pivot away from the term sewage, go to better words, wastewater or use water or clean water. So we just changed it to selectivity.
It's clever, of course. In Icarus, they got too close to the sun. In your case,
and in our case, that's what we're hoping to avoid. Yeah, crash and burn.
Right. What else? I think I've summarized most of the questions here. There are a lot of particular ones about the effect on the clarifier or the effect currents in the clarifier. I think we did address that. Is there any potential for a biological degradation of the membranes themselves? So the membranes themselves potentially biodegradable, or
because everything's potentially biodegradable, or at least it'll deteriorate it. We have not looked at that part. I mean, a PVDF membrane that we were looking at is pretty tough, but the issue here is normally the membranes don't see the light of day, and here they're while they're not directly facing the sun, they are exposed to ambient solar radiation and other weathering things. So that is something. And actually, James Lund has a question related to sunlight penetration. I mean, so yeah, I mean that's another thing we need to fine tune. The material that you make the pot out of. We use plexiglass, but you want something that's basically UV durable. I mean, you have high UV transmittance, obviously, but you want it to be durable and not break apart. So that can, maybe again, it's a sort of optimizing. You might have a material that's very good for that, but then now the cost will go up, versus if we use a cheaper material that will break down under UV, but hey, maybe it's very cheap, it's okay.
Yep.
But then at that point,
James Lund also asked, are you aware of the revolving algal biofilm technology developed at Iowa State University by Doctors Wen and Gross? Have you seen that?
Yeah, actually, I think I met them actually when I visited Iowa State as part of the CAPI lecture. Yeah, their process is pretty cool. It's basically you're just constantly harvesting the algae as you're growing and generate biomass. So I think that's a great way, and the economics work out for them. I think there you have to be not so picky about the algae, because it's going to be a mixed culture when it's exposed to the environment. But if you're just after biomass, it might just be good enough for what you're doing.
Yeah, it brings up work done by others. I don't know if you're also aware of work done by Professor Christo Delatos at my institution, Stevens Institute, on growing algae attached to surfaces. In his system, he flows the wastewater over a surface in a shallow bed and grows attached algae, which is then removed by a mechanical scraping approach. Different approaches, different physical approaches.
Yeah.
Okay. I didn't read everybody's questions directly. I hope that my rephrasing and condensing has satisfies everyone's questions. If there's anything, if you type into the Q&A right now, a question that you think didn't get answered adequately, we'll get it to them immediately. Otherwise, we'll sort of watch get close to wrapping this up. Just give it a few seconds to see if anything shows up. And nothing has. I think with that, Dr. Ye, I think I'm just going to say thank you very much. This has been really interesting. I hope everyone enjoyed it. I see that the vast majority of the participants has hung on right to the end to hear everything that you had to say. So again, thank you. At this point, I will hand it back over to Marissa for some concluding messages.
Okay, thank you everyone for all your questions, and thank you to Dr. Ye for sharing your knowledge with us today. We have several webinars planned in the near future, so you can go to aaes.org/events and reserve your spot. Just a reminder, if you're not yet an AAES member and you're considering joining the Academy, please email me to discuss our membership options. My email is up here on the screen right now. And last but not least, the PDH certificates for this event will be sent out sometime this week. That's all for now. We enjoyed being with you all today. Thank you so much and have a great day.
Thanks for having me. Okay.